Welcome back, everybody. Hope you had an awesome time learning about passive transport in the last lecture. So today, or this time, we're going to talk about active transport and then move on to some other important parts inside of the cell. So we're still in chapter 3, and let's get those slides open so we can begin. And hope you have your coffee. I actually don't, but you need it. So get your coffee or your tea or your chocolate milk or whatever it is that you use while you're taking your notes. Okay, so we left off right here talking about plants and why they'll wilt if you don't water them. So water your plants. And now we're going to talk about active transport. So we talked about passive transport, both diffusion and osmosis. Diffusion is the movement of the particles from an area of high concentration to an area of low concentration. And osmosis is the movement of water from an area where the particles are in low concentration to where the particles are in high concentration to try to dilute them out. Okay, sometimes cells need to move things in or out against that gradient, right? So let's say there's only a little bit of potassium inside your cell, but your cell wants more. And your cell wants more than is outside, right? It doesn't want it to be even inside and outside the cell. It wants all the potassium, right? It's going to have to work for it. These things are, right, diffusion happens naturally to make things even. If the cell wants things uneven, it's going to have to put a little effort in. So active transport is using energy to move molecules in and out of cells. So a classic example is this sodium-potassium pump that cells use to move potassium out of the cell and to move sodium out of the cell and potassium into the cell. You guys don't need to know which is going in which direction. That's not the important part. The important part is to understand the active transport. So if we look at this picture, you can see the sodium concentration. That's the one in red here. Na is sodium. The concentration is low inside the cell, right? This is in, this is out. And it's very high outside the cell. All these orange sodiums are getting pumped out. The cell does not want sodium inside of it. If it left it up to diffusion, it would be even on both sides. And the cell wants a lot of potassium, so eat your bananas. The potassium is very high inside the cell, right? Here we can see it's very high, and it's low outside the cell. Any potassium, the cell is pumping into it, right? This uses energy. It's not happening naturally. So that's active transport. And so this will take place in one of those special protein channels. Just like the water moved through a channel, you can see that the sodium and the potassium are moving through these channels. They're not just going to diffuse through the cell membrane because the cell needs to work on it. It needs to pump it. Okay, well, what if something is too big to fit through these channels, right? Not everything is as small as these little protein, secret passageways that the cell sets up. When there's something too big, the cell is either it's going to have to take it in or get rid of it in a different way. This also uses energy. Okay, this is also active. Endocytosis or exocytosis. So endo means inside and cyto is cell. So this is taking things inside the cell. And exo means outside, like an exoskeleton on a bug, right? So this is moving things outside the cell. And the way this works for endocytosis, and you guys don't need to learn the kind of smaller terms for whether it's a solid or a liquid that's getting moved. But if this is your membrane in yellow here, and this big red thing the cell wants to take in, it just kind of engulfs it in the cell membrane and pinches it off, right? So here's your cell membrane, and it pinches off something, and then it closes up. Okay, and now we have this thing that we wanted, this big red particle is now inside the cell. And then this cell membrane can open up, dissolve, and you've got this red thing in there now. It can do it with, big particles. It could do it with these smaller things, or like liquids is what we have here with the pinocytosis, right? So it wants some of these blue things that couldn't get through on their own, and it just engulfs it in a membrane. Exocytosis is the exact opposite. Cell wants to get rid of all these little green things, right? It engulfs it in a membrane, and that membrane then attaches to the cell membrane. It opens up and sends them out. Okay, so that's endo and exocytosis. It's just using that membrane system to transport things in or out. Okay, so we've actually moved through a lot of chapter 3 here, right? We talked about the basic cell, right? We talked about prokaryotes and eukaryotes and some of their differences. We've just talked about cell membranes and how things get across the cell membrane. And now we're going to talk about some of the other organelles inside of a cell, the little organs. So the main ones, we're not talking about every single thing in a cell. Your book will have more, but you're not tested on things that I don't cover it in lecture, okay? We're going to talk about the cytoplasm, that's a jelly inside. That endomembrane system, when we talked about endosymbiosis and how infolding had created a nucleus and some of those other, the endoplasmic reticulum. We talked about those very briefly. We're going to discuss now in detail what these different systems do. And then we'll talk about cell walls, which are different from the membranes, and chloroplasts, okay? So we'll go through things one at a time. Cytoplasm, so cyto is cell, as we've talked about, and plasm is like plasma, right? If you have like a plasma TV or something like that, it's a jelly. So cell jelly is just a jelly that's inside every single cell. So if we're looking at a picture of a cell and in lab, you look at cells under the microscope, like this, and here's the nucleus, and then this everywhere I'm coloring in blue just looks empty. It's not empty, it's not filled with air. That's actually cytoplasm. You just can't see it. It's clear jelly, okay? Inside that clear jelly, though, are microscopic, so, or smaller than microscopic. We can't see them with our types of microscope. There actually is a skeletal structure in there. It's not bone, obviously, but it's proteins, little tubes that actually add a support system to the cell, so the cell doesn't get totally squished. And it can enable it, it can give it a little bit of movement. If you think about if you had like a balloon that you were trying to roll, if there were some spokes or something in there, right, it would have a little more support to move. And that's true for cells as well. Okay, let's move on now to this endomembrane system. So these are multiple organelles, but what's in common is that they are bound by membranes, just like the plasma membrane on the outside of the cell. So remember, we talked about how this likely evolved by having this outer membrane and then bits pinched off, right? And then another bit pinched off, right? So things pinched off, and we have this endomembrane system now, an inside membrane. And these do a variety of different things. So we're going to walk through them one at a time. So the first endomembrane inside a cell is the nucleus itself. For the nucleus, in eukaryotic cells, remember prokaryotes don't have a nucleus, this membrane around it is actually the exact same lipid bilayer as outside cell membrane. Okay, it's the same thing, it's just inside. This is the control center. Everybody probably knows what the nucleus is, right? It's kind of the headquarters. The DNA is inside there, so it's storing all of that DNA. And it has some kind of landmarks even inside of it. So if we're looking under the microscope, sometimes you'll see some of these things. So it has this membrane. It has the thin DNA, that's the chromatin, and then you'll see, sometimes you'll see a dot even inside of the nucleus. That is called the nucleolus, and that makes ribosomes. So we're going to talk about ribosomes in a minute, okay? So those actually then get transported out, and they're going to be outside of the cell. Even prokaryotes have ribosomes. Everybody needs ribosomes. They make proteins. So those are a really important part of a cell. In a eukaryote, which we're talking about here, eukaryotes are the only ones with nuclei. They're going to send those ribosomes outside, and they're going to get stuck onto one of those other membranes. So here we've got a picture of the nucleus. Here's our nucleolus. making ribosomes. You've got your DNA, which they're calling chromatin here, same thing. Ooh, okay. And then we see these other membranes stuck on the DNA. It looks like a maze that they've set up, right? So this is actually all of these blue maze pieces are called endoplasmic reticulum. These little red dots, these are the ribosomes that were being made in the nucleolus, and they got shipped out, and now they're stuck on the endoplasmic reticulum. So we're going to talk now about the endoplasmic reticulum then, because I'm sure you're like, well, what does it do? I see it here, and I see it with ribosomes on it. Tell me more. So I will. So the endoplasmic reticulum that has ribosomes stuck all over it is called the rough endoplasmic reticulum because it's bumpy because it has all those ribosomes. Okay, if it didn't have the ribosomes, it would be called smooth. This one's rough. You can also just call it rough ER if you don't want to call it endoplasmic reticulum. It is this series of membranes that are just all folded up, stuck on the nucleus. Okay, so we've got our nucleus here, and we've got our endoplasmic reticulum there. And all over it, it has, that picture was showing it in red, so they've got all these little ribosomes on it, okay? Its primary function here is the ribosomes are making the proteins, and so then this endoplasmic reticulum is going to fold them up and send them out. Remember how when we were talking about proteins, you had your primary structure of amino acids, and then they got folded up with their hydrogen bonds, and they had a secondary structure, and then they may have a tertiary structure where they're all specially folded up. That's what your endoplasmic reticulum is doing. It's folding it. Okay, they're like the origami experts. So here we have the rough ER. Here's just another picture. In this instance, the ribosomes are in yellow, these yellow dots. Outside of the rough ER, there's smooth ER. It's same folded sacs. It doesn't have ribosomes, so this isn't doing anything with proteins, but it has different jobs. And here we've got another picture of rough ER. Beyonce dressed as one. I think this is like the Met Gala or something one year. Okay, so again, you see these folded up sacs with the ribosomes on it. Okay, if you're not rough, you're smooth. Okay, so the smooth ER, that same look of that membrane all folded up, but it doesn't have ribosomes. So it's not doing anything with proteins. Instead, it is making fats actually and steroids, and it's detoxing cells so it can remove toxins. So you can think about this, different cells may have different amounts of rough and smooth ER. Ooh. And I'm going to write that down. That is going to be, I think, our question. Why? Which cells might have more rough ER or smooth ER? Now that you know a little bit about their jobs. Okay. Now the next part of this membranous system that we're going to talk about is the Golgi apparatus. This is also, another membrane inside of the cell. This is taking all those things that were made packaged up by the rough and smooth ER, so lipids, proteins, and it's going to package them, right? So if the smooth ER did the folding and now we need to ship it out, okay, let's put it inside another membrane. Let's, send it to the right place. This is basically sometimes, thought of, if you remember your like 9th grade bio, they say like this is like the UPS center, right? So they're going to package stuff up. Same with the lipids. If the smooth ER made the lipids, send it off to the Golgi apparatus and it's going to package it up and send it out. They can also make carbohydrates, okay, so it can help build some things. And this is made of 4 sacs, so we'll see here. Okay, so if you look at this, obviously this is a drawing, but if we look at this basic architecture of the cell, the layout makes sense, right? You've got the nucleus in the middle. It made the ribosomes here inside the nucleolus. It shipped them out. It makes the proteins and folds them up in the rough ER. It makes the lipids and sends them out in the smooth ER. Then everybody just sends it over to the Golgi apparatus. It's going to package it up, right? So we go from nucleus to these either rough or smooth ER, and then to the Golgi, and that's now the closest thing to the edge of the cell, and then it can send things out. So it's, you know, it's kind of an assembly line. It's got this conveyor belt system, and it's well organized. Now the final part of this endomembrane system we're going to talk about is the mitochondria. This is also made up of these sacs, but remember we talked about the fact that the mitochondria likely evolved from endosymbiosis, right? So a larger cell engulfed a smaller one. So this is inside living. Living is the bio, and together is the sim, right? Inside living together. It took that mitochondria in. The mitochondria could already convert food products into energy. Cell energy is ATP, okay? This little cell was doing it. A bigger cell said, come on in, why don't you work for me, right? I'll give you a place to live and you can do my energy conversions for me. And now they're together. ATP requires converting that carbohydrate to ATP requires a lot of oxygen. So most of the oxygen that we breathe is for use by mitochondria. Again, some cells may have a lot of mitochondria, some cells may not have as much. Active cells are going to have a lot more mitochondria in them than less active cells, like fat cells. They don't have as many. So every single cell in your body doesn't look the same. They may have different numbers of organelles, one kind or another. And again, they have their own DNA. So you can actually study mitochondrial DNA and sequence that, and it looks very, very different than the genomic DNA, the DNA that's in your nucleus. So here's a picture of mitochondria, and it does have an outer membrane and an inner membrane, and kind of another matrix set up in that, just kind of like the endoplasmic reticulum did. We're going to talk about mitochondria and this energy conversion, I think, in the next chapter. So you'll learn a little bit more about how it's organized inside of there. But it is a membrane system in there. Okay, so that, I think, is our last membrane part of the cell. And now we're just going to talk about a few more kind of landmarks and organelles that some cells have. Not all. Cell walls are an important structure for some organisms. So, and some of the organisms that have cell walls are eukaryotes. Plants are eukaryotes. Fungi are eukaryotes. But bacteria are prokaryotes. I'm sorry, my handwriting there is bad, but you can write bacteria down if you want. So the cell walls are not found evenly across like the living world, right? So some eukaryotes have them, like plants, and you can see those plant cell walls under the microscope that we've looked at in lab. And some prokaryotes have cell walls. Cell walls are just giving them a little additional structure. So if you think of, here's our cell membrane, it could be like a water balloon, and then you could put that inside of a box, right? You could put it in a cardboard box. It's giving it a little more structure. It's a little easier to package your boxes on top of each other if you wanted to build a stem, right? Than to package your little, blobby cells on top of each other. So this enables some plants to go really, really tall. because they have that cell wall giving them a little extra structure. And they are made of cellulose and lignin for plants at least. They're cellulose. For fungi, it's lignin. For bacteria, it's another structure again. So these are different carbohydrates. So unlike the cell membrane that's made of a lipid bilayer, right, the cell wall is made of a carbohydrate. Okay, and then one other, I think our last structure that we're going to talk about inside cells are chloroplasts. So these are only in eukaryotes because this is an organelle. It's not a cell wall, which is outside of the cell. This is something inside of it. Prokaryotes don't have anything inside except for some DNA and ribosomes, right? They don't get all these special little organs. The chloroplast is found in eukaryotes that can photosynthesize. So this means it's in plants, and it also is in some algae, which are protists. Algae like seaweed and stuff, they're actually not plants, okay? So plants and eukaryotic algae are the organisms that have chloroplasts. And this is where photosynthesis takes place. So this is where they take sunshine and they're able to convert it into carbohydrates. They make sugars, okay? The chloroplast is what does that. And again, this likely evolved through endosymbiosis, the same way that mitochondria got into a larger cell and started working for them. The chloroplast likely did the same thing. It was a small photosynthetic organism, got engulfed by a bigger organism, and now they work for it. Okay, so they this is the site where photosynthesis takes place, and we're going to learn more about photosynthesis in the next chapter as well. But here is a drawing of a chloroplast. Again, you have these membranes, right? This was like the cell membrane of that little photosynthetic organism that got eaten up. And then inside of it, there are these stacks that look like stacks of green pancakes. These are thylakoids, and we'll talk about all of those in more detail in the next chapter. And then to end with, I just have a little chart here for you guys of the different parts of the cells that we talked about and whether they're in prokaryotes and animals and plants, which are eukaryotes, both of these. So the plasma membrane outside the cell, everybody's got them. Cytoplasm and cytoskeleton, everybody has it. The nucleus, only eukaryotes have a nucleus. Ribosomes and rough ER. So the prokaryotes actually have the ribosomes, yes to ribosomes, but no to the rough ER. I should have divided those two out. Mitochondria, only the eukaryotes. A cell wall, that some prokaryotes have them, some eukaryotes do, some don't. So that's not evenly distributed. Chloroplasts, only plants, and then also algae, which are a type of another type of eukaryote, okay? But they're just not on this chart. The Golgi apparatus, again, only eukaryotes. Smooth ER, only eukaryotes. Flagella and cilia, we looked at those at the very beginning of this chapter, right? Those are, flagella is a tail, and cilia are little like hair-like things. Some for everything, we'll say some. For some animals, the only flagella are on sperm. So in humans, males have some cells with flagella, females don't. For plants, they have eggs and sperm too. So the sperm of plants will have flagella, but the other cells won't. Some bacteria have flagella in order to move around also. So that, again, just depends on the specifics. Okay, so that is the end of chapter 3. And you can go now into your discussion questions. Bye-bye.